Today's topic is translation, and you're very familiar with this slide. You have seen it multiple times. You know what split genes are now, the definition of introns and exons. You have a broad idea of what a unit of a gene is. You do know that genes can take up a lot of space in the linear double-stranded helix, which folds into a chromosome. You know that they are very large genes, they are very small genes, but the gene size has nothing really to do with the size of the mRNA because there are a lot of introns. You know what the process of transcription is, and you know what a pre-RNA is, and you know that there is an event called splicing, Nobel for splicing, Nobel Prize for transcription, Nobel Prize for split genes, which Philip Sharp and others, which I haven't talked about. And you finally get a piece of RNA with leader sequences and lagging sequences, which finally is translated into a linear amino acid chain. The 5' to 3' basically leads to a N-terminal to C-terminal amino polypeptide chain. And this polypeptide chain has an ability to compact itself, not stay linear, into a folded state, which we call as a protein. And over the years, we have solved many protein structures, of folded protein structures, using primarily X-ray crystallography, but also a second technique, which became popular in the 90s, which is nuclear magnetic resonance. So let's start with a simple view of translation, which gives you an overall idea. And then I'll fill in all the details in the next few slides.
Translation is the second stage of protein synthesis where a piece of mRNA is used to create a polypeptide chain. Firstly, let's go over the key terms. Remember that mRNA or messenger RNA is made up of codons. tRNA or transfer RNA has an anticodon on it, and the anticodon defines the amino acid carried by the tRNA.
So this is a fairly important point which I haven't really introduced for the non-biologists, that the sequence on the mRNA is read as a triplet. That is, 3 nucleotides at a time define a code, and these 3 nucleotides over here define one code, and the next 3 and the next 3 and the next 3. This is something which is very familiar to people who have done biology. So what this movie is going to focus on is, of course, it will talk about the ribosome, but I'd like you to pay attention to how following molecules, the mRNA, the transfer RNA, which is carrying an amino acid, it's a carrier of amino acid, how the codon and the anticodon recognize each other, and how, as you will see, this leads to protein synthesis using the mRNA as a template.
Translation takes place in the cytoplasm, and the first thing that happens is that the ribosome attaches to the mRNA at the start codon, which is AUG. The ribosome is made up of ribosomal RNA, rRNA, and ribosomal protein, and has 2 sites on it, the P site and the A site.
Now, these are just arbitrary nomenclature. In this particular movie, the lady who's speaking will define the P site as a parking site. That's not official nomenclature. And the A site is the so-called attachment.
A tRNA carrying the complementary anticodon to the start codon, which is always AUG, comes and attaches in the P site. The first amino acid of this is always methionine. A piece of tRNA having an anticodon complementary to the mRNA's second codon then floats in from the cytoplasm, carrying the next specific amino acid. In this case, that's tyrosine, and this tRNA then attaches in the A site. A peptide bond is then formed between the first 2 amino acids. At this point, the tRNA that's in the P site, or you might want to think of that like the parking site, is no longer really required because its amino acid is attached to the amino acid on the second tRNA by the covalent peptide bond. The ribosome then moves along the mRNA by 1 codon. This places the second tRNA in the P site, and the first tRNA is then free to break away. While it does so, a new tRNA with an anticodon matching the mRNA's codon and carrying a specific amino acid can then come and join in the A site that's been freed up. Consider this an attachment site. A peptide bond then joins this new amino acid to the existing chain. And so this process continues. The ribosome always moves down by 1 codon, and once it's done so, it means that the previous A site is able to break away free from its amino acid and allow a new tRNA with a new amino acid to come and join in the A site that's freed up. The new amino acid can then join the existing polypeptide chain with a peptide bond, and the tRNA that was freed up goes back to the cytoplasm and collects another specific amino acid ready to be used again. Note that the amino acids used are specific and defined by the codons on the mRNA and complementary anticodons on the tRNA. The process is somewhat repetitive and continues until the ribosome encounters a stop codon on the mRNA. The stop codon does not code for an amino acid, but merely indicates where the process of translation is terminated. Once the process has been terminated, the polypeptide chain, tRNA, ribosome, and mRNA detach, and the polypeptide chain becomes free-floating in the cytoplasm. Thus, in translation, overall, we have used the mRNA to form a polypeptide chain.
All right. So,
Translation is
hopefully, what this movie does, and one of the reasons I show many of these movies, is they are the simplest representation of translation without too many added complications. And this movie gets to the heart of the matter, which is basically the fact that the mRNA is used to basically information in the mRNA is translated to information in the protein. There is a three-letter codon. The codon is recognized by an entity called as the transfer RNA. The transfer RNA at one end has what is called as an anticodon. The codon and the anticodon recognize themselves. Very critical in biology, this recognition. The tRNA is carrying an amino acid, and the complementary codon-anticodon recognition allows it to allows the mRNA to specify which amino acid is going to come first, which next. The sequence of 20 amino acids, one at a time, is defined by reading the mRNA by the ribosome, which is the translational machinery. So what I'm going to do in the next 3 or 4 slides is now focus on each of the components and near the end show you a slightly more complicated molecular picture of how translation takes place. Now, before I go ahead, are there any questions at this point about this movie per se? Any terminology questions, for example? Let's go over the different elements. The first is, of course, this large macromolecular complex called as the ribosome. And I have already told you a couple of times that it was a big surprise when researchers realized the ribosome is not actually a protein enzyme, it is an RNA enzyme. So the ribosome is a mixture of proteins and RNA. We call this RNA, which is part of the structure of the ribosome, and of course, which is the enzymatic active entity, as ribosomal RNA or rRNA. Now, rRNA is very distinct from the tRNA, which you saw in the last movie, and I'll talk to you about tRNA also. Fine. To give you an idea, now these are, of course, the actual structures of which is the atomic level structure solved by X-ray crystallography of the 70S ribosome. Now I'll define what the 70S ribosome is, but basically what you're seeing over here are different angles of looking at the same structure of a bacterial E. coli ribosome. Okay, it has 2 units, and this is a simplified version. There's a large subunit and there is a small subunit. And in order to execute protein synthesis, these 2 subunits have to sit on, well, the large subunit has to sit on the small subunit. Okay. Now, you'll also realize that when these 2 subunits come together, many years of molecular biology with a lot of experiments done blind mostly have made scientists realize that they can define 3 sites. The A site, which is somehow not clear over here, the P site, which you have heard of, and there is also an E site. And the simplest definition of E site is an exit site. Okay. So you saw in the movie before how the P site and the A site are kind of important. You'll realize that the mRNA goes through this large ribosomal complex at the point in which the small and the large subunits meet. So the ribosome is basically going to cross over here. Okay. You'll also realize, and this will become very obvious in movies, that the actual ligation will happen somewhere over here where I've drawn in red. And now I'll just change it to blue. And the protein will basically come out over here, N terminus to C terminus, from the top of the large subunit. Fine. So in a single slide, I'm giving you a broad picture of what the large complex looks like, its shape. I'm showing you actual X-ray crystallographic structures, showing you the A site over here, the P site over here, and the E site. I know you won't be able to interpret them. Even researchers who have actually not doing crystallography cannot look at these structures and easily interpret them, which is why on the right-hand side, you have a simplistic representation. I've also put, for extra reading, this is not mandatory for all of you, a review by Venki Ramakrishnan, written in 2002. And a few years later, 5 to 6 years later, Venki Ramakrishnan got a Nobel Prize for solving the structure of the ribosome. Okay, so these are the elements. For those of you not used to terminology, again, you have to pick up all this terminology of E site, A site, P site, large subunit, small subunit, nothing more complicated than that. So this now introduces you to one major player in the translation in making proteins. Now, a little bit about the structure and function of ribosomes, and as always, I keep on telling you that you should have a historical timeline in your mind. There was a lot of work in the 50s, 60s, 70s, 80s, 90s, 40, 50 years of work. The ribosome was a very, very challenging problem simply because it was a very large complex made up of somewhere between 50 to 100 different independent units, and these units would be either RNA or protein. The size of the ribosome ranges from around 20 to 40 nanometers in diameter. It's really very, very large. Much of the initial work was done on prokaryotic ribosomes, and we use the term 70S usually to define the prokaryotic, or more specifically the E. coli ribosome. And the generic eukaryotic ribosome is called as the ATS particle. Now, S is basically a unit of mass, just like Dalton's is. And it's based on how heavy a large molecule, because we are dealing with a very large molecule over here, is in during centrifugation. And I will not really go into a definition of all of. The size of the eukaryotic ribosome is 4 into 10 raised to 6 Daltons. You know what Dalton is, a unit of mass. And the prokaryotic ribosome is approximately 2.7 into 10 raised to 6 Daltons. Really very large complexes inside the cell. Now, in this schematic, you see that there are 3 major RNAs and in the prokaryotic ribosome and 4 major RNAs in the eukaryotic ribosome. Okay. Now, don't be taken by this very simplistic picture of one for prokaryotes, one for eukaryotes. On the right hand side, as you can see, there is a range of ribosomes, which from E. coli all the way to Homo sapiens, and their sizes, shapes vary. So there is a lot of variation in ribosomes. This is what we have discovered in the last 20 years. Also, to remind you that mitochondria and chloroplasts are inside eukaryotes, and the origin of these mitochondria and chloroplasts is actually a prokaryotic cell. So the mitochondria have their own independent ribosome, which is very distinct from the prokaryotic ribosome and the eukaryotic ribosome. And these ribosomes are functional, and in the mitochondria and in the chloroplast, protein synthesis takes place just like it takes place in the eukaryotic cytoplasm. Now, the Nobel Prize for events related to structure and function of ribosomes was given to these 3 scientists, Venki Ramakrishnan, Tom Stites, and Ada Yonath. All 3 of them contributed in different ways, but much of their contribution, for example, Ada, Thomas, and Venki were all crystallographers. They were also biochemists. They did a lot of biochemical studies. And as I said, there's a lot of wet biochemistry over here. Purification of these 50 to 70 units, each unit had to be purified and a ribosomal complex formed that had to be crystallized. And even the solution of the interpretation of the X-ray pattern was not trivial. This was one of the most challenging problems in the last 30 or 40 years. Okay, now let me move on to another aspect of what we are talking about, that there is a genetic code, universal genetic code, which exists in all plants and animals and prokaryotes, which basically defines the relationship between the 3-letter codon on the mRNA and the way it is interpreted and converted into an amino acid. And many tables on the internet. This is just one of them. And what this does is it basically says that if you have a 3-letter code, there are 64 possible combination of triplet codons. Okay, but they aren't 64 amino acids. In nature, there are only 20 main native amino acids. Okay, they are non-native amino acids, which we will not talk about in this particular class. And the way the scientists who worked out this code, and I will define who these are in the next slide, worked it out, was that you have a 3-letter codon, but it's the first 2 letters which are very important. So this is the first position, this is the second position, and this is the third position of a 3-letter codon. So if we take any one, let's say we take U. So U and U is phenylalanine. So if you have a U on an mRNA, that basically means that during the translation process, the transfer RNA, which has an anticodon equivalent to UUU, will carry phenylalanine or leucine in this case. So you have UUU is phenylalanine, UUC is phenylalanine, UUA is leucine, and UUG is again leucine. So if it is UUC, there'll be an anticodon on the tRNA. It will recognize the mRNA codon, and the phenylalanine amino acid will be placed first in the P site and then in the A site, and it will be ligated to the next amino acid in sequence. All right. So this table basically defines the relationship between the codons and the amino acids, and this was established by work by Nirenberg and Khorana and others, which I will basically talk to you in the next slide. Methionine, which is AUG, functions as both as a start codon and as an addition of methionine, which is AUG, and UAA and UAG function as stop codons, as does UGA. one very clear thing which comes out of the solution of the genetic code done by the researchers who are there in the next few slides is that the third position has a so-called is more flexible than the first two positions, right? So for example, phenylalanine has two options, UUU and UUC. Proline, for example, has four options, CCU, CCC, CCA, CCG. So the third codon has what we call as a wobble or flexibility, which is part of biology. We don't completely understand why this is so. And this also leads to degeneracy in the genetic code. Are there any questions on this slide? Okay. So now the genetic code basically relates a triplet codon to an amino acid, and it is very critical for the whole process of translation. And the understanding, the cracking of the genetic code. Remember, we are talking about 1968 when the prize was given. Much of the work was done between 1961 and 1965, though of course all work has previous work on which these researchers based on their research on. And this was barely a decade after deciphering the structure of DNA. And remember, in the 1950s also, people didn't really believe that DNA was the genetic material. And the cracking of the genetic code in the 60s and the Nobel Prize awarded in the late 60s basically brought home completely clearly that at least in prokaryotes, where much of the work was done, that there was a very clear genetic code. Okay, so here are the three people: Robert Hawley, Hargobind Khurana, and Marshall Nirenberg, who were given credit for cracking the genetic code. Hawley basically was the person who discovered, sequenced, and found the role for transfer RNA in this process. Hargobind Khurana was a chemist who did the first synthesis of nucleotides. And this was a very important step because if you synthesize nucleotides and you put it in an extract which contains a ribosome, then depending on what kind of nucleotides you synthesize, for example, I'll give you an example up there. As I told you, UUU was phenylalanine. If you make a nucleotide which is UUU, just a series of U's, and then you put it inside an extract with a ribosome, and you get only and you look at what polypeptide is synthesized, and you find out that the only amino acid which you see is Phe, right? You realize that this triplet code probably relates to phenylalanine. And because you can now synthesize oligonucleotides, done for the first time by Hargobind Khurana, you can now put any sequence you synthesize of different combinations and look to see which amino acids are being made by the ribosome. Thus, you can then have a relationship between the nucleotide sequence and the amino acid sequence. So Nirenberg was the first person to sequence the basis and define each codon, and help, of course, by work done independently by Hargobind Khurana. And Robert Hawley brought in the concept of transfer RNA. So he discovered that part of the protein synthesis process. For undergrad students, you should be aware of a program called as the Khurana program, run by the Government of India, University of Wisconsin-Madison. They support undergrad students to move to basically go to a university in the US for internships. So you can look up that. You can try and apply for the Khurana program in the future.